So we're at the National Physical Laboratory in Teddington, just on the edge of Bushy Park. And the reason it's significant is because the particle sampler that you can see over my shoulder featured in the study that we have conducted with the collaborators on this exciting project. So just to illustrate, this is the kind of filter that goes in our devices. So you can imagine, it's very simple, the air is drawn through and particulate matter that's airborne gets deposited on that filter. That filter is removed from the device after whatever period of sampling, and that's all very standardized and regulated, and then that will be analyzed for its contents. So what you're looking at is, in this case, a sort of thin filter made of, in this case, a cellulose fibre-based material. We had never analysed them for environmental DNA and hadn't considered it.
And so they contacted us to ask whether it was possible that the same process they were already using was capturing DNA in the same way that we were attempting to do it. And we had no knowledge of their networks of air samplers, but because we spoke with them extensively about how they operated, we collectively got the idea that they may be capturing the very material that we were trying to filter ourselves.
We keep those samples for a year. And the reason why we keep them for a year at all is because we might have to go back and do a repeat measurement on a filter just to check that the measurements are okay. So it was completely fortuitous that we had a year's worth of data from which we could select samples to send to our biology collaborators, and from which they could then do their eDNA studies. So my understanding is that what we've effectively gathered is cellular material, cells that have been sloughed off by animals. You can imagine birds flying around, they're flapping around, they're shedding material. There's a whole manner of dispersal mechanisms by which these cells are released into the atmosphere, and then we literally capture them on these filters.
There are small regions of DNA, very tiny pieces, that can be used to distinguish animal life very efficiently. We use different targets for different taxonomic groups sometimes, so a different region for plants than we would for animals. But you really only need some of these very, very tiny regions to help you distinguish between species. The major questions that we need to answer now is how far away can we draw that material, and how long does it last in the air? And that will be influenced by things like weather and wind and climate and where you physically are. And those are all open questions. This is a pilot study to show that these samples that are being collected anyways may have extraordinarily important biological value to us as ecologists. The type of material that we are targeting and collecting would tell us the species. We know there are humans, we know that there are birds of different types and different types of mammals and different types of trees. But these are really tiny degraded fragments that we are capturing. We also take measures to try and suppress our human signal in our analysis, because there's just so many of us, we would easily swamp the signal. The more we look for environmental DNA, the more we find it. It's carried around on the leaves, on trees. You can wash them and get DNA from animals that have passed by. It's in the soil. It's carried around on us, material from other organisms, because it's really just everywhere. What we're trying to do is find better and better tools to capture it, to learn more about it. It's effectively free information there that we can try and use for conservation.